Welcome to the deep dive. Today we're tackling a really big topic, general anesthesia, or GA.
Mm.
And if you've ever thought it's just like flipping a light switch off and on for consciousness, well, our sources today show it's way, way more complex than that.
Oh, absolutely. It's this incredible journey, really, through how drugs work in the body, neurobiology, even how brain networks behave on a large scale.
So our mission here is to pull out the key insights from, well, a pretty hefty stack of research.
Yeah, we're looking at basically three big things. First, how clinicians figure out the right drug dose when patients are so different, physiologically speaking.
Right, the whole pharmacokinetic challenge.
Exactly, PK/PD.
Yeah.
Second, mapping what actually happens in the brain when you lose consciousness and then, crucially, regain it.
And third,
And third, this fascinating idea that falling asleep under anesthesia and waking back up, they aren't just mirror images of each other. They're distinct processes.
okay, let's start with that first challenge then, the huge variability between people. Propofol is kind of the star player here, right? It's known for working fast,
Super fast acting.
but why does it wear off so quickly, even if someone's been under for hours?
Yeah, it's a classic pharmacology question, really. It comes down to distribution kinetics. Propofol is extremely lipophilic.
Meaning it loves fat tissue.
Exactly.
Hmm.
So when you give that initial dose, the drug floods out of the blood really fast and gets soaked up by peripheral tissues like fat and muscle. That quick movement out of the central circulation is what makes the effect stop so fast initially.
You mentioned capacity, and the sources talk about VDs, the apparent volume of distribution at steady state. Can you unpack that a bit? Why is that so important for a drug like propofol?
Right, VDs. It sounds complicated, but think of it like this. The body acts like a giant sponge for propofol because it dissolves so well in fat. The VDs can be huge, like 3 or 4 times the actual physical volume of the patient's body.
Whoa, okay.
Yeah, it doesn't mean the drug is literally floating in that much fluid. It just means the body's tissues, especially fat, hold on to a massive amount of it relative to the blood. So when you stop the infusion, that sponge effect pulls the drug out of circulation almost immediately. That's your rapid offset.
Got it. That explains the quick wake up. But okay, how do you build a single mathematical model for dosing when you've got tiny kids at one end and morbidly obese adults at the other? The standard hemodynamic per kd approach must just fail.
Oh, absolutely. It falls apart. Researchers figured out pretty quickly that simple linear scaling doesn't work. You need different ways to describe different parts of the process.
Like what?
Well, for instance, total body weight TBW turns out to be the best way to predict the volume the drug spreads into because, more body, more sponge.
Okay.
But interestingly, fat-free mass, basically lean body mass, is a better predictor for how quickly the body clears the drug metabolically.
Huh. So you need two different weight measures just for one drug's model. How do they even combine that into something usable in the clinic?
They use something called allometric scaling. It's a way of relating biological factors to body size using a power law, not just a straight line.
Power law.
Yeah, instead of just multiplying by weight, they use weight raised to an exponent, often 0.75. Sounds technical, but it basically allows the model to bend and adjust much more accurately across that huge range of body sizes, from really small to really large.
Okay, that makes sense. And did these big studies, like the leveled one mentioned, find any specific patient factors that really matter when using these refined models?
They did, yeah. Some really key things came out. First, the size of those peripheral compartments, the sponge parts we talked about, that volume V2, it actually gets smaller as people get older.
Interesting. Okay.
Second, metabolic clearance, how fast the body breaks down the drug. It's higher in women than in men.
Ah. So that explains why clinicians often say women wake up faster from propofol.
Precisely. It provides a solid pharmacokinetic basis for that clinical observation. And finally, weight itself, specifically the central compartment volume, it scales differently. It follows more of a sigmoid or S-shape curve.
What does that mean for dosing?
It means the induction dose per kilogram should actually be relatively high for very low weight patients, but then comparatively lower for very obese patients. You don't just keep increasing the dose per kilo indefinitely.
Wow. So it's really personalized medicine, dialing in the dose based on age, sex, and exactly where you are on that weight curve? Okay, that's the drug delivery. Now, what happens when that perfectly calculated dose hits the brain? We shift from PK models to brain networks.
Right. Now we're talking neuroscience. Researchers use tools like fMRI to look at functional connectivity, or FC. Think of it as mapping the brain's internal communication highways.
And what happens to those highways under deep anesthesia?
This is where it gets, I think, really fascinating. One of the big findings is that deep GA, and this was shown for both propofol and the inhaled anesthetic sevoflurane, it causes a kind of collapse of the brain's uniqueness. They call it a loss of brain fingerprinting.
Wait, loss of individuality, like the specific way my brain talks to itself just fades?
Exactly that. When you're awake, the map of your brain's functional connections, your connectome, is pretty stable and very distinctively yours. You can identify someone just by looking at it.
Okay.
Under deep anesthesia, those individual patterns become much less unique, much less self-similar over time, and much harder to tell apart from anyone else's connectome. It's like everyone's brain activity starts to look more generic, more similar.
Is that happening everywhere in the brain, or does it affect certain areas more?
It's definitely not uniform. The effect is strongest along what they call the sensory association axis. So the higher level thinking areas, the default mode network, the frontoparietal networks involved in attention and control, they lose their distinctiveness much more than the basic sensory processing areas, like the parts dealing with touch or vision.
So the networks involved in like self-awareness, planning, abstract thought, those are the ones that lose their unique signature first. That feels significant.
It really does. And there's this amazing evolutionary angle, too. The studies found that under anesthesia, human brain connectivity patterns, when you look at them in a sort of simplified mathematical space, they shift to become more similar to the patterns seen in macaque monkey brains.
Wow.
And the specific brain regions that show the biggest drop in this humanness are precisely those parts of the cortex that expanded the most during human evolution. It strongly suggests anesthesia temporarily dismantles the most complex, recently evolved layers of our conscious architecture.
That's incredible. It's not just turning the volume down. It's like temporarily reverting to a more fundamental, less individualized state.
Okay, so that's going under. What about the other side, waking up? You mentioned it's not just the reverse process.
Yeah, the sources are really clear on this. Emergence isn't just induction run backwards. And figuring out why is super important if we want to develop drugs that can specifically help people wake up smoothly.
How do we know they're different? What's the mechanism?
Well, we know natural sleep and wakefulness are controlled by specific brain circuits, right? Like the ones involving the hypothalamus that von Economo identified way back. And aesthetics seem to interact with these natural pathways.
So how does the brain tell the difference between being knocked out by a drug and actively waking itself up?
A key player seems to be a group of neurons called orexinergic neurons located in the lateral hypothalamus. They're absolutely vital for staying awake.
Okay, orexin wakefulness promoter. Got it. Right. Now, the surprising thing is research shows these orexin neurons selectively control emergence from anesthesia, both inhaled and IV agents. But, and this is the kicker, they don't seem to regulate the induction, the process of going under.
Hang on, that feels backwards. If orexin keeps you awake, wouldn't blocking it help put you to sleep? Are you saying it's only involved in opening the door back to consciousness, not closing it?
That's exactly what the evidence suggests. If you give extra orexin, it helps speed up emergence. They're even looking at plasma orexin levels as a potential biomarker for predicting wake-up times.
Huh.
But it gets even more specific depending on the drug. For propofol and sevoflurane, fiddling with another area, the paraventricular thalamus, or PVT, affects both going under and waking up.
Okay.
But for isoflurane, another common inhaled agent, the PVT seems to only modulate emergence.
The level of detail is just amazing. A tiny difference in thalamic involvement depending on the specific anesthetic used.
And we can zoom in even more. For propofol specifically, the induction part seems linked to boosting GABA activity in a small spot in the brainstem called the pontine reticular formation, the PNO. That specific molecular tweak seems to kick off the whole process.
Which forces us right down to the molecular level. For years, everyone learned the Meyer-Overton rule, basically. The better an anesthetic dissolves in fat, the more potent it is.
The lipid solubility theory, yeah.
But now the thinking is it's mostly about specific protein receptors, right? What are the key targets the sources identify?
Yeah, the focus has definitely shifted to proteins. For propofol, its main job is activating the GABA A receptor, which is the brain's primary off switch, its main inhibitory system. It doesn't just generally muck with the receptor, it has a very specific binding spot involving an amino acid called methionine 286 on one of the receptor subunits, the beta subunit. It's like a key fitting into a lock in the locus coeruleus, a key arousal center.
A very specific molecular interaction. What about isoflurane, the inhaled one?
Isoflurane's main target seems to be different. It primarily inhibits voltage-gated sodium channels, NAV channels. These are crucial for nerve firing. Isoflurane binds directly to the channel protein and changes how it responds to voltage, making it harder for nerves to fire repeatedly.
So direct protein hits for both. What does that mean for the old lipid theory? Is the idea that anesthetics just dissolve in the cell membrane basically dead, at least at clinical doses?
That seems to be the consensus now, yes.
Yeah.
Recent studies looking at volatile agents like isoflurane found they didn't really change the general physical properties of the lipid membrane itself at the concentrations used in surgery, like 1 to 2 MSC, minimum alveolar concentration.
Okay.
You only started seeing significant changes in the membrane lipids at crazy high toxic levels, like 12 to 18 MSC. So at therapeutic doses, the action really appears to be direct binding to proteins like those NAV channels, not just messing up the whole membrane.
Right. A major shift in understanding. Okay, let's touch on xenon. It's unique, this noble gas anesthetic, because it also seems to protect neurons. How does it pull off that double act?
Xenon's got a really complex profile. For the anesthesia part, it does a bunch of things. It blocks excitatory glutamate receptors, specifically NMDA and AMPA types.
Mm.
At the same time, it enhances certain potassium channels like TREC1 and KTP, which helps calm nerve cells down. Plus, it dials down activity at a couple of other receptors, NACHR and 5-HT3.
So it's hitting multiple targets to induce anesthesia
Huh.
and the neuroprotection side.
That seems to be a separate mechanism activating cellular survival pathways. Xenon actually increases the levels of pro-survival proteins inside the cell, things like BCL2, ACTP, CREB, and BDNF, which promotes neuron growth and survival.
Okay.
And simultaneously, it decreases proteins that trigger cell death, like BAX and caspase-3. Importantly, this protective effect isn't just passive. It depends on active cell processes like calcium signaling and making new proteins. It's like xenon actively tells the cell to switch into survival mode.
Fascinating. Okay, we've gone from dosing models down to molecules and back up to networks. Let's bring it back to the patient experience waking up. After someone's been under for a while, say with isoflurane for a few hours, what comes back online first, cognitively speaking?
Yeah, this is another area where the results were pretty counterintuitive. The sort of standard assumption maybe was that basic functions would come back first, and then the really complex executive functions handled by the front parts of the brain would be the last to recover. Simple reaction time before complex problem solving.
Right, you'd think abstract thought would be the foggiest, longest lasting effect. What did they actually find?
They found the opposite. Executive function things measured by tests requiring abstract matching or concept formation. It actually recovered surprisingly early and got back close to the person's pre-anesthetic baseline pretty quickly, both in terms of speed and accuracy.
Seriously, so you might be able to think logically quite soon, even if other things are still off.
It looks that way. The functions that showed the most prolonged recovery, the things that stayed impaired for longer, were tasks tapping into basic processing speed, sustained attention, and simple reaction time tests, like the psychomotor vigilance test, or PVT, and motor praxis tasks. Your brain might be thinking relatively clearly, but your ability to react quickly or stay focused is still lagging.
That's wild. What was the brain activity doing during that time? Did the EEG data match this pattern?
It did, yeah. They looked at brain complexity using a measure called permutation entropy, PE. They saw complexity dropped when consciousness was lost, as you'd expect. But crucially, it returned to baseline levels just before the person actually started showing behavioral signs of waking up, like opening their eyes or following commands.
Okay.
And right after emergence, the complexity of the frontal lobes was significantly higher than in the back of the brain. This strongly suggests a kind of frontal-led recovery of brain dynamics, which lines up perfectly with that quick return of executive function we just talked about.
Wow. Okay, this deep dive really underscores that GA isn't simple at all. It's dynamic, incredibly individual, and definitely not just an on-off switch. We've seen the need for sophisticated, personalized dosing.
Using things like allometric scaling and specific patient factors, yeah.
That waking up is its own unique neurobiological process, maybe involving systems like orexin.
Absolutely, not just the reverse of induction.
And that, maybe surprisingly, our ability to think abstractly might recover faster than our basic reaction speed.
Exactly. And for me, perhaps the most mind-bending takeaway is that finding about functional connectivity, that deep anesthesia temporarily dampens down the unique communication patterns that make your brain.
Making us all more similar, functionally speaking.
Right? Erasing that individual fingerprint, making human brains even look functionally a bit more like macaque brains in some ways. It really makes you wonder, doesn't it? If our higher order consciousness and individuality are so tied to these complex long-range network patterns, What else could studying functional connectivity, especially under different states, tell us about the nature of consciousness itself, maybe even across different species?
A really provocative thought to end on. What is the fundamental network basis of consciousness and how far does it extend? Lots to think about there.